All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, process of recombinant DNA technology. In this, isolation of DNA. Second, fragmentation of DNA. Third, isolation of desired DNA. Fourth, ligation of foreign DNA to vector. Fifth, introduction of recombinant vector into host cell. Sixth, culturing of host cell for large scale. Seventh, obtaining gene product. The first, isolation of DNA. Undoubtedly, nucleic acids are genetic material in all organisms without any exception. DNA genetic material in most of the organism. And DNA should be isolated in a purified form in recombinant DNA technology. Why it should be isolated in a purified form for recombinant DNA technology? Because the experiment is going to be done with restriction endonucleases, which act on only purified DNA but not associated with other macromolecules like carbohydrate, protein, or lipid. So it should be obtained in a purified form. Next, as the DNA enclosed within the membranes, DNA present in the chromosome present in nucleus, surrounded by nuclear membrane, in turn surrounded by cytoplasm, which in turn surrounded by nuclear membrane, in case of plant surrounded by cell wall. In animal cell, just with membrane. Next, as DNA intertwined with histone protein, non-histone protein, also RNA, carbohydrates and lipids. So by appropriate methods and enzyme, you should free from other macromolecules like protein, carbohydrate, RNA and also lipid. So you must use the enzyme proteases to digest protein, carbohydrates to digest carbohydrate, RNases to digest RNA, lipases to digest lipid, but not DNases because we require the DNA. DNases hydrolyze the DNA into deoxynucleotide. So we should not use DNases. Next, it is subjected to centrifugation, and to precipitate the purified DNA, we have to use a precipitant, chilled ethanol. So purified fine threaded DNA precipitated out, present in a suspension, and by using glass rod by stirring motion technique, the purified fine threaded DNA taken out. As DNA is sticky, it adheres to the glass rod. By stirring motion technique, the fine threaded DNA forms a spherical ball around the glass rod. What do we call this step? It is spooling. The spooled DNA ready for fragmentation, that is restriction enzyme digestion in next step. So the purified DNA undergoes fragmentation by using restriction enzyme. Such restriction enzyme digestion require the purified DNA to be incubated and optimize the condition for restriction enzyme. Agarogel electrophoresis checks the progression of restriction enzyme digestion. As the DNA is negatively charged, move towards the positive, that is anode side. And the source DNA and vector DNA cut with the same restriction enzyme. So the cut desired gene in case of from the source and cut gene from it is vector in a space, both are mixed and added with the DNA ligase, recombinant DNA molecule formed. In next step, the desired gene to be amplified to several copies, several billions of the copies. It may be done with the source as well as vector also. The technique it is polymerase chain reaction. In polymerase chain reaction, the steps which are denaturation, annealing, extension. The requirement, a thermocycler, a computerized machine which maintains thermal cycles, and two sets of oligonucleotide, that is deoxynucleotide, and DNA polymerase, that is tech polymerase, obtained from Thermus aquaticus, that is thermostable enzyme that remain active even during temperature induced denaturation of DNA. Tech polymerase active only at high temperature, that is going to be wrong. It remain active even during high temperature denaturation of DNA. Next, genomic DNA acting as templates. So, two strands of the DNA unwinded at high temperature, that is denaturation. Next step, that is annealing. Two sets of primer, that is the two primer deoxynucleotide attached to third end of each template. So primer attached to third end of each template, what we call hybridization, annealing or cooling. Next step, third step, extension. Which are extended? Primers are extended. As primers attached to third end of the template of the DNA on both strands, tech polymerase adds deoxynucleotide to third end of the primer. So which got extended? Primer got extended by tech polymerase by adding what? Deoxynucleotide already present in this. So DNA synths occur in 5-3 direction added to third end. So it is amplified to several billion copies. So 1 billion copies of the DNA obtained after 30 cycles. And this polymerase chain reaction was discovered by Carey Mullis. Next, introduction of recombinant DNA into host cell. Plant, animal, bacterial cells, after making competent to accept the recombinant DNA or foreign DNA, the recombinant DNA inserted into the host cell. If the recombinant DNA bearing antibiotic resistant gene ampicillin, it should be plated on a medium having antibiotic ampicillin. Only transformed cells will survive, non-transformed cells would die. So in this, ampicillin is said to be a selectable marker. And there is a question with regard to this biotechnology in exercise 9th question. Can you say answer the reporter gene or reporter enzyme monitor bacterial transformation with a foreign DNA in addition to selectable marker? The selectable marker, which are antibiotic resistant genes like ampicillin, which cannot differentiate recombinant and non-recombinant. But for survival of the cells, the antibiotic resistant gene it is required. For screening of recombinant, what is required? That is reporter gene or reporter enzyme. Reporter gene, LAGZ gene, reporter enzyme, beta galactosidase. The reporter gene not only confirms the transformation, but also distinguish from recombinant from non-recombinant. Because recombinant colonies which are colorless white, non-recombinant colonies are blue in color. Next step, obtaining foreign gene product. The alien piece of DNA introduced into the vector, that is inserted into the vector, now it is recombinant vector. Thus recombinant vector transferred to host cell, plant, animal or microbe, where the alien piece of DNA gets multiplied to several copies. So ultimate aim in all recombinant technology is to produce the gene product, that is the protein. To ensure the alien piece of DNA, the gene to express to produce the protein, it requires optimized condition, some technical condition. So we provided everything, we optimized the condition. Then you have to think of it producing in large quantity. Why you have to think of it producing large quantity? Because you provided everything to meet the demand. So large quantities of the protein are expected. Next, the protein encoding gene expressed in heterologous host, what do you call it? Is recombinant protein. The cells harboring the cloned gene of interest, that is In case of small volume cultures, that is laboratory method, the cultures are used to extract and purified by different technique. This is the small volume cultures in laboratory method for certain products. Cells can also be multiplied in a continuous culture system. In continuous culture system, large vessels, the bioreactors are used, where the inlets as well as outlets. In continuous culture system, used medium drained out. Fresh medium is introduced from one point. So, to ensure the cells in active, physiologically active, exponential log phase to produce higher biomass, high biotechnological product, that is the protein. Because small volume cultures do not yield appreciable quantities. So for large volume cultures, 100 to 1000 liters are processed in a container, stainless steel vessel. What do you call it? Is bioreactor. So bioreactor is a stainless steel vessel where raw materials are biologically converted into products by using microbial plant animal cells. So products are formed in case of bioreactor. But these bioreactors require optimized condition, temperature, pH, oxygen, nutrients. So everything should be provided in case of the bioreactor. Next, there are the two types of the bioreactor: simple stirred tank bioreactor, sparged stirred tank bioreactor. In simple stirred tank bioreactor, it is cylindrical, curved at the base. Such a symmetry ensures the mixing up of the content. Suppose it is a flat vessel, there is no mixing up of the content. And stirrer, a rod with the blades and a mortar, the stirrer continuous circulation. The stirrer facilitate even mixing oxygen availability throughout the container because it is not static. Continuous stirring by a rod and blades and a mortar. The stirrer facilitate even mixing oxygen availability throughout the container. And this there is an agitator system. The agitator system is nothing but a rod with the blades and a mortar. Continuously it is agitating in order to provide even mixing oxygen availability. And there is a oxygen delivery system and foam breaking system. Upper blades meant to control what is the foam. Lower flat blade impeller conserves circulation of what is broth. And pH controlling system, temperature controlling system, and there are ports. The ports are the places which are inlets and outlet where samples are periodically collected for testing in simple stirred tank bioreactor. So to carefully observe the diagrammatic representation and the labeling, it is very, very important. And this question, the labeling with regard to agitator system, appeared in 2025. Next, sparged stirred tank bioreactor. In sparged stirred tank bioreactor, sparging, forcing of air. The forcing of air create air bubbles. The bubbles increase oxygen transfer area. So there is increased transfer area for oxygen and there is a gas entrainment. So further there is a large production because of availability of it is oxygen. Next, downstream processing. Suppose it is the initial process, the first step, the middle one, the biotechnology product obtained, and the last it is marketing. So after obtaining biotechnological product, before marketing as a finished product, separation and purification, collectively what do you call it is downstream processing. Up to this obtaining biotechnology product, it is upstream process. After obtaining this product, before finished product, separation and purification collectively called downstream processing. The product has to be subjected to formulations. That is, you are going to add certain preservatives to keep in a viable state for long time. After formulations with certain preservatives for preservation, it should be subjected to, that is, clinical test in case of the drugs. Quality control testing is applicable to all. So, whether veterinary product, food product, or agricultural product. So, quality control testing and downstream processing varies from product to product. It is not the same in all products. I am signing off from this episode. You will see in case of the next episode, biotechnological application. If you feel the approach is right, don't forget to subscribe and share to the persons those who are seriously preparing for NEET. Listen with concentration, don't skip, don't scroll.